For my next presenter, I'm going to welcome to the stage, make sure I have my right notes here. Thank you, Dr. David Herrmann. So Dr. Herrmann is the E. Philip and Carol Saunders Professor in Neuromuscular Research, and he's also the Chief of the Division of Neuromuscular Disorders at the University of Rochester. His research focuses on inherited peripheral neuropathies, including new gene identification and efforts related to clinical trial readiness, which is what you're going to hear about today, including leading the Accelerate Clinical Trials for CMT study, which is also known as ACT CMT, and you can see the logo for that right here. Without further ado, Dr. Herrmann.
Thank you very much, Steve. Well, all right, thank you. I just need to determine how to advance the slides here. I see. So thank you very much for inviting me to speak today. And I'm going to be talking on the topic of clinical trial readiness, why this is important and really urgent now, and a very specific aspect of it, the importance of natural history studies in preparing the field for future clinical trials. And we've heard about the urgency of being ready for trials in humans, because we know that as researchers, we've got really very proficient in curing and treating animals with CMT, and many different types of CMT, be it CMT4C, CMT1A, CMT1X, mitofusin-2, CMT2A. In animals, they're effective treatments. Many of these are genetic treatments. And we've heard from Dr. Cortese and others the amazing story of the discovery of SORD deficiency neuropathy, and that has gone very rapidly from an animal model and discovery now to a human trial. But for many other types of CMT, we're a little further behind, but we need to be ready to conduct those clinical trials. So at the most simple level, what we want are approved treatments for different forms of CMT. And we heard realistically that despite our best efforts, most trials are not successful. We always learn a lot from many trials, but we do want to improve that success rate. And so to improve that success rate, we obviously need the correct treatment approach. And we learn a lot about the correct treatment approach from all the work that happens in different animal models, because if we can treat animals successfully, at least we know that we're targeting probably the right part of the mechanism or pathogenesis of the disease. But in translating it to humans, a human is very different from an animal model, and we have to be able to design the trials correctly. Because if we think about treating humans, it's not sort of like treating pneumonia, where we can rapidly determine whether a treatment's working or not, and we can see the effects of treatment. We know that for most types of CMT, CMT is very slowly progressive. And realistically, we're not going to expect to give an injection of a gene therapy or give a patient a pill and see treatment responses very rapidly. And so we have to really, there's a whole science in designing trials correctly and appropriately to be able to pick up small effects of treatments that may initially be small effects, but we hope over time and years of treatment may actually end up being large, important, life-altering effects. So what I'm going to talk about are the efforts to really learn how to design clinical trials correctly. And the FDA is very interested in this topic, and they have a guidance document to pharmaceutical companies on the topic of natural history studies for drug development. And that will be my focus of the discussion today. And so firstly, I'm using this term natural history studies. Many of you who have participated in clinical research may have actually participated in a natural history study and may not be entirely aware what it is, why is it important. But firstly, a natural history study is really getting at the natural course of a disease that a disease takes in the absence of any intervention. What is the normal or natural progression of different types of CMT? And studying that is really the essence of a natural history study. So natural history studies typically follow a group of individuals over time who either have or are at risk of developing a specific medical condition. And by studying individuals and following individuals over time in very specific ways who have a particular form of CMT, we can learn if there are genetic factors, environmental factors, and other factors that impact the course of disease. And this is all very important for us as we think about developing clinical trials. So how do you run a natural history study? Well, there are two different broad types. There are what we call retrospective studies, and retrospective studies are the most basic form of natural history study. Here, the data that's analyzed is data that's already been collected. Very often, in a confidential, de-identified way, one can go to the medical charts or medical records of patients with a particular condition, in this case CMT, who may be living across the world, and there are examples of this, and one can do a systematic analysis in the context of a protocol and confidentially to learn a lot about the CMT, how it progresses, and its manifestations. And all that information is very important for actually designing clinical trials. But a more rigorous way to conduct a natural history study is what we call a prospective natural history study. Here, there's a protocol that's developed, and patients with CMT would enroll in the natural history study. And then as a group, they're observed over time. They may be evaluated every 6 or 12 months for a defined number of years. And the data collection for natural history studies can be in person at research or clinic visits, or they may be remotely now in many instances using telemedicine. And what sort of data is collected in a natural history study? Well, the data that's collected is patient-reported information. Patients may actually complete specific questionnaires that give us insights into what patients are experiencing, what the impact of CMT is on the individual. It may also include neurological examinations or physical function assessments, and also evaluations of various biologic samples that we term biomarkers, and I'll speak about those a little bit. So what's the value of these natural history studies? Well, in this graphic from the FDA, they help us design clinical trials. Very often, by following patients over time, we can get clues to how to treat disorders. So they may actually give clues to particular therapies that might be useful from the information we actually learn by studying the disease in detail. They also define the target population for clinical trials. Who should participate in a clinical trial with a particular type of CMT? At what age? What severity of CMT might be suitable to participate in a clinical trial? And by following individuals in natural history studies, we can also learn how to best track progression of disease. And for a very slowly progressive disorder, this is really very, very important. We have to be able to precisely measure whether the CMT is progressing and how it's progressing, and do that in a practical way. So for this, using these natural history studies, we can develop what we call clinical assessments or clinical outcome measures that may be the way that we determine in a trial whether treatments are working. So the FDA indicates that ideally, when they approve a new treatment, it either improves how individuals are functioning or how they're feeling. And so we have to develop standardized assessments of how individuals with CMT are feeling and experiencing their disorder, but also how they're functioning. And additionally, natural history studies can help us develop biomarkers, which are biologic measurements that give us insight into CMT and its progression. So to do this work and to conduct these natural history studies, approximately 14 years ago, a consortium of researchers across the world, now more than 20 centers, was developed called the INC, or Inherited Neuropathies Consortium. And the work of the Inherited Neuropathies Consortium has been supported through a grant from the NIH Rare Disease Clinical Research Network. Researchers have worked together to study many aspects of CMT, but one aspect has been the conduct of natural history studies. And so what do we know about the history, natural history of CMT? Well, a lot of what we have learned has been used through using a scale known as the CMT-NS or Charcot-Marie-Tooth neuropathy score. And many of you may, if you've been for a clinic visit or been participated in a study, actually had the Charcot-Marie-Tooth neuropathy score assessed. It just takes a few minutes to assess. Involves answering 3 symptom questions. It involves 4 examination elements where your sensation is assessed for pain sensation with a pinprick, your ability to feel vibration sensations assessed, and then strength in the arms and legs is assessed. And then for the full scale, there are 2 nerve conduction studies that are just done in 1 limb, and we call that the CMT-NS. If we omit the nerve conduction studies and just do the clinical elements, which most of us are more comfortable with, we call it the CMT exam score, CMT-ES. And a lot about what we know in terms of tracking CMT has come from assessing the CMT exam score in individuals with CMT over time. And we've learned a lot about the natural progression of CMT from that. So as an example of a retrospective natural history study, the Inherited Neuropathies Consortium, and this was led by colleagues in Italy initially, studied the natural history of a very rare form of CMT called CMT4B. And this is an area, there was discussion yesterday, where there are likely will soon be clinical trials. There's a lot of treatment avenues. And there are probably just a few hundred individuals in the world who have CMT4B. And yet, by conducting a natural history study through a confidential chart review that actually spanned the globe, they were able to identify 50 individuals and learn a lot about, using the CMT exam score, learn a lot about CMT4B, how it presents, and possibly how it progresses. And this information will be very useful to design the clinical trial. Who should participate? How long should that clinical trial last? How do we actually pick up treatment effects? In more common forms of CMT, the Inherited Neuropathies Consortium, through our Rare Disease Clinical Research Network centers, has learned about the natural history of CMT1A, the natural history of CMT2A, and through the work of Dr. Friedman, has led work in understanding the natural history of CMT1B. And in work by Dr. Record, we've learned about the natural history of CMTX, so the more common forms of CMT. And what is in common for all of these, although some progress at faster rates, is you'll notice on the x-axis of all these graphs, follow-up is in the matter of years. So we see slow worsening over time. All these trend lines are trending upwards, which means higher CMT exam scores, but that's happening over multiple years. And so what we learned is if you just were to track CMT with a CMT exam score, you would have to follow individuals for a very long time in a clinical trial to pick up small treatment effects. And that may not be practical. And hence the field has really worked on developing different ways through natural history studies to assess progression of different forms of CMT. And another example of this is CMT4C, which shows a very similar pattern of slow progression over time. So what are the other ways we can measure CMT? Well, researchers within the Inherited Neuropathies Consortium have developed functional assessments for children, a CMT infant scale, a CMT pediatric scale. And using these, we've been able to study the natural history of CMT in children. And we know that across different forms of CMT, with the CMT pediatric scale, we can pick up a 14% worsening in physical functioning over a 2-year period. So when trials are conducted in children with CMT, particularly faster progressing forms, it may well be that the CMT pediatric scale, which is an upper limb and lower limb and gait and balance and strength assessment, will be used to determine whether treatments are working or not. My colleague at University of Rochester has developed a very similar measure called the CMT Functional Outcome Measure. And the elements of it are shown if they project, but this is assessing upper limb function with tests such as a functional dexterity test, a test looking at putting pegs into different holes on a board, strength evaluation in the upper limbs, gait and balance assessments, and other evaluations. And this is developed for adults, but it's very similar to the pediatric scale, except it's more appropriate for adults. Similarly, we've developed ways to understand more systematically what patients are experiencing. So for adults, we've developed something called the CMT Health Index, where many of you may have completed this, but it's a questionnaire where there are questions that are designed to understand what the impact is of CMT on different aspects of one's day-to-day life. And we hope that these measures, which we're now studying over time in different subtypes of CMT, will allow us to better determine in clinical trials whether treatments are having a positive impact. Now, what about this term biomarkers? Many of you may have heard the term biomarkers, why they're important, how we're studying them in natural history studies. Well, a biomarker is simply a biologic marker in blood, other body fluids or tissue that can be an indicator of the state of CMT. And they may be used to tell us how well the body is responding to treatment. But why, if we have all these assessments of function and questionnaires, why are biomarkers important? Well, we hope that they may provide an earlier indicator of a response to treatment than a patient's symptoms or examination. Because CMT is very slowly progressive, we would ideally like to be able to give a treatment or a genetic therapy and know within a few months whether we are seeing early impact of that treatment. It may be that the treatment only shows clinical benefit or functional benefit a year or two later, but really for practical purposes for drug development, we need to know soon whether treatments are working or not. And there are various types of biomarkers, and I'll discuss some of the ones now that have been developed through natural history studies. So we've heard a lot about MRI of leg muscles developed by the University College of London. And if you look at this image, you can see the panel. The top panel to the left shows a healthy muscle with a lot of muscle. But the panel at the bottom shows smaller muscle where most of the muscle is replaced by fat. And the fat in your muscle has nothing to do with the body fat that each of us has. They're unrelated. But really, if you want your muscle to work well, we cannot have fat replace the muscle inappropriately. So it has been shown that in patients with CMT, there is more rapid accumulation and replacement of muscle by fat in the lower limbs. So one way that we hope is that by giving a treatment, it can slow the replacement of muscle by fat. And by tracking MRIs over time, there have been a lot of natural history studies now across different forms of CMT. This may be one way to detect treatment effects. Is a treatment slowing the accumulation of fat in muscle? We at University of Rochester have looked also at the sensory nerves. CMT is, of course, a motor disorder with atrophy and foot drop. But important also is sensation. And so we have a technique called confocal microscopy of Meissner corpuscles. These are the little sensory nerve endings in the skin that allow you to feel touch. And you can see to the right under the panel that says control, the arrows point to these little sensory nerve endings in the skin. But as your Charcot-Marie tooth neuropathy score gets worse, so you lose those. And so in a multi-center study that we call CMT now, we're actually doing the Meissner corpuscle imaging on individuals. It's painless. It just takes a few minutes. We look at it in the hands, and we're tracking these sensory nerve endings to see how they, what happens to individuals, numbers of those over time, and whether it might be useful to use that as a way to determine whether treatments are having a positive effect. I have a colleague at the University of Rochester, Dr. Herrmann, is also looking at muscle ultrasound. And so to the left, you can see a big healthy muscle of a shin muscle from a patient, but to the right, you can see a muscle from an individual with CMT1B. The muscle is smaller and looks different. And this work is just at the beginning stages, but across different types of CMT, we're looking to see whether ultrasound of muscle could be an effective way to detect treatment effects. What about blood? We heard Pat Livney and others talk about, can't we all give a tube of blood? And I do really believe that simplicity is important. And I do believe in our plasma, our blood, the solutions do lie there for us to understand what's happening with CMT. So what is plasma? Well, if you separate out the blood shown in red, the red blood cells, the rest of that is plasma. And then we can look for chemicals or molecules in blood. One of them I've shown you as a study of something called neurofilament. And the hope is that we can identify, and there's a lot of work that's gone on now to identify molecules in blood that potentially might indicate the severity of the CMT, and that if you give a treatment, you may rapidly see an improvement in that biomarker in the blood, and that then may predict later clinical improvement. So there's a lot of work being done to discover biomarkers in plasma. And finally, my colleague, Dr. Kate Eichinger at University of Rochester and Josh Burns have looked at something called wearable sensors. And we have studies, natural history studies, where we're following patients with different types of CMT over time. In children, adults, CMT1A, 1B, CMTX, CMT2A, and others, using wearable sensors. And most of us have sensors on. I've got my Apple Watch on, I've got my iPhone in my pocket, and these instruments are collecting a lot of data. I don't go to the gym without knowing exactly what is happening. But there are more sophisticated, precise sensors. So one type of sensor is called an actigraph or physical activity monitor. And we're conducting within the Inherited Neuropathies Consortium natural history studies using these. An actigraph is worn around the waist for a week. You often go to your doctor's visit or your research visit, you're given those, and it allows us to monitor your activity with CMT in your home and community environment. Because ultimately, when we do treatment trials, one way we might be able to know whether we're having a really important effect is if activity levels increase. So with an actigraph monitor, we can see how many steps you take, how much time are you engaging in different activities, and what's the intensity of your activity, and how much time do you spend sitting? Not meant to be good for us. So that's one type of activity monitor. The other one is something called an APDM gait and balance monitor. And we do this in the clinic, and we do this over time, every 6 or 12 months. And here you can put sensors on the chest, the arms, the waist, the ankles. They're very lightweight. And then you can ask patients with CMT to walk 10 meters, walk for 6 minutes, do a balance task, and you can get a lot of information beyond simply timing how long it takes to do those. You can get information about how much you're swaying when you do a balance task, how long your step length is, your step speed, and all of these parameters. And why do we do this? We're hoping with this non-invasive way that one day we can use these in clinical trials to pick up very early on whether one's gait or balance is improving before one might notice this. And we use these natural history studies where there's no intervention to really track all of this. So many of you may have participated in natural history studies. And I think the key, Dr. Zuckner and the geneticists were talking about, we need to find ways to engage so this is accessible for everyone. Just like every person with CMT should give a tube of blood, and we need to find a way to have their genome sequenced, we also need individuals to be able to participate. So, we learned some ways of finding clinical trials to participate in the studies of the Inherited Neuropathies Consortium. You can go to this URL, and there are a number of sites within the US. And there are other sub-studies that by reaching out to individual sites or reaching out to us at University of Rochester, we can try and match you with studies, ongoing natural history studies. All of this, we hope, will very soon allow us to design clinical trials well, and hopefully there will be many of those for individuals to participate in. So thank you.